Good day to everyone. I'm Sheila Marvi Mangyang from the Group 4. And now I will going to discuss or report the chapter 9, which is the gene therapy. But first, let me define the learning objectives. At the end of this unit, the students are able to first discuss the concept of gene therapy and its various forms. Second, discuss and assess the benefits and detriments of gene therapy to human health. And lastly, assess the issue's potential benefits and detriments to global health. Human body is composed of trillions of cells, and cells are the basic building blocks of all living things. Genes are regions of DNA that code for proteins, which are molecules that build, regulate, and maintain the body. And every person has two copies of each gene, one from each parent. Sometimes there is a change in genes' DNA sequence, such as a substitution, deletion, or duplication, and this is what we call mutation. Now, let's define gene therapy. So, gene therapy is a technique that modifies a person's gene to treat or cure disease. It is a technique that uses genetic material for the long-term treatment or genetic disorder. They use gene therapy because it has the potential to treat disease that cannot be treated with conventional medicine. Some of these diseases are partially or fully caused by genetic mutations. And this is the example of cardiovascular disease, vision disorders, diabetes mellitus, and etc.
Good day everyone, my name is Wency P. Maralit from BPED 4C. And today I will be going to discuss the approaches to gene therapy. Gene therapies can work by several mechanisms. First is replacing a disease-causing gene with a healthy copy of the gene. Second, inactivating a disease-causing gene that is not functioning properly. And last is introducing a new or modified gene into the body to help treat a disease. Let's proceed to types of gene therapy. There are two different types of gene therapy depending on which types of cells are treated. First is somatic gene therapy. In somatic gene therapy, the somatic cells of a patient are target for a foreign gene transfer. In this case, the effects caused by the foreign gene is restricted to the individual patient only and not inherited by the patient's offspring or later generations. Somatic cell gene therapy would aim to cure a disease only in the patient, not in the patient descendants. It was initially conceived as introducing a properly functioning copy of a gene into a person who had a genetic disease as a result of inheriting only improperly functioning copies. Last is germline gene therapy. In germline gene therapy, the functional genes which are to be integrated into the genomes are inserted in the germ cells. Example, sperm or eggs. Targeting of germ cells makes the therapy heritable. Germline gene therapy will correct the genetic variants of the reproductive cells of an individual, and this would be passed down to future generations. This therapy removes a hereditary disorder from a family line forever. Hereditary disorder occur at humans are possibly inherited from the germline cells. However, curing this disease is possible only through modifying their nuclear and mitochondrial DNA mutations in preimplantation embryos, which is commonly known as germline gene therapy. That would be all, and let us hear more from our next reporter. Thank you.
Good day everyone, I am Rona A. Ritotal from BPED 4C and I'm going to report the gene therapies strategies. And there are 4 strategies and these are the gene augmentation therapy. The second one is the targeted killing of specific cells. Next is the targeted inhibition of gene expression. And lastly, the targeted gene mutation correction. Okay, I will only discuss the first two, which is the gene augmentation therapy and the targeted killing of specific cells. The first one is the gene augmentation therapy or GAT. In GAT, simple addition of functional alleles is used to treat inherited disorders caused by genetic deficiency of a gene product. Example, GAT has been applied to autosomal recessive disorders. Dominantly inherited disorders are much less amenable to GAT, and here is the example of GAT. A gene therapy vector has been designed to treat the disease cells with a gene X. This vector was introduced inside the disease cells by various gene transfer methods. After a successful homologous recombination, the treated cells will show the presence of gene X products as well as normal. The second one is the targeted killing of specific cells. It involves utilizing genes encoding toxic compounds, the suicide genes or prodrugs, regions which confer sensitivity to subsequent treatment with a drug, to kill the transferred of transformed cells. This general approach is popular in cancer gene therapies. And here are the two direct killing of disease cells by two methods. The first method is the introduction of toxin gene into the disease cells, which mean expresses toxin protein, the cells die. The second method, prodrug gene, it involves incorporation of a certain gene. Example, the TK, in the gene therapy vector which shows a suicidal property on introducing certain drug. The example of this is the gang-C-clovir. Lastly, the assisted killing of disease cells by immune system cells. Assisted killing is another strategy of killing disease. Here, one method is to insert well-known foreign antigen coding gene which induces immune cells for the killing of the disease cells. Few more methods are base immune cells activation in which a certain cytokine encoding gene incorporated into gene therapy vector and inserted into either disease cells or non-disease cells. This will lead enhanced immune response followed by killing of disease cells. That will be all. Thank you.
Good day everyone, I'm Floridine B. Umiles, and for today's video, we're going to continue the gene therapy strategy. First is targeted inhibition of gene expression. This is to block the expression of any disease gene or a new gene expressing a protein which is harmful for a cell. This is particularly suitable for treating infectious disease and some cancer. So what is target gene expression? The target gene expression is analysis with qPCR aims at the quantitative measurement of mRNA expression of defined target genes. Example in different genotypes prior to enduring drug therapy or after the administration of compound candidates in the preclinical phase of drug environment. Gene inhibition or gene inhibition is a therapeutic approach that involves detecting or silencing the expression of a mutative gene that is not functioning properly, or 1 to 3. The gene inhibition works by preventing gene expression at the post-transcription level, thus RNA or gene inhibit gene expression. RNA are the direct products of genes, and these small RNAs can be detect or direct in same conflicts to degrade messenger RNA or mRNA molecules and thus decrease their activity by preventing translation by post-transcriptional gene silencing. As you can see in figure 4, to inhibit the target gene expression in disease cell, the antisense mRNA coding gene inserted vector or triplex forming oligonucleotides or TFO or antisense oligonucleotides or ODAN can be introduced which will inhibit the gene expression either by forming DNA or RNA triplex inside the nucleus or forming. So let's go to the last one, or targeted gene mutation correction. It is used to correct a defective gene to restore it for function, which can be done at genetic level by homologous recombination of at mRNA level by using therapeutic ribosomes or therapeutic RNA editing. So as you can see in figure 5, this is used to disease caused by mutation. The correct gene will be swapped by the mutant gene X, then decrease cell by becoming normal after the correction of the mutation by gene therapy. Targeted gene refer is a technique used to correct a mutation at a specific site in an episome or chromosome. It uses synthetic oligonucleotides together with the cell inherent DNA refer system to direct single base by pair changes, a type of mutation in which a chromosome gene is altered by the substitution of a DNA construction assembled by in vitro. In mouse, the constructs are usually designed to imitate gene functions such as targeted mutation are often casually referred to as knockout. That would be all. Thank you very much.
Good day everyone, I am Kim Soral Quintorubia, BPED 4C. For today, I will be discuss the methods of gene therapy. The goal of gene therapy research is to determine whether a new gene can be used to replace or inactivate a muted gene to treat a disease or help the body fight a disease. Gene therapy is currently an experimental treatment that replaces or inactivate disease-causing germs through injection or IV. Research continues into the ways this therapy can treat cancer and other conditions. For a gene to be delivered into a cell, there is a transporter is typically used. A transporter is known as the vector. So let's discuss first what is a vector. A vectors are essentially vehicles designed to deliver therapeutic genetic materials such as a working gene directly into a cell. Vectors are typically derived from viruses because viruses have proven to be very efficient at finding their ways into cells. In order to make vectors safe to use, all of the viral genes are removed, and the vectors is modified to deliver only the therapeutic genes. A vector is made from an altered virus because the virus is used as a vector. Its viral genes are removed. Once the virus is modified, it is intended to transport the desired gene to a cell without causing disease. Once inside, the desired gene should restore the function of the protein. Vectors can be given intravenously, which means they are administered into a vein or injected into a specific tissue of the body. Other procedures, such as surgery, can also be used to deliver vectors into specific areas of the body. There are mainly two approaches for the transfer of gene in gene therapy. The first one is transfer of genes directly to cell into the body or called the in vivo, and the second is transfer of gene into patient cell outside the body or called ex vivo gene therapy.
Good day everyone, I am Mary May Rosalyn S. Nuevo from Group 4. And now, let's talk about the ex vivo gene therapy. Ex vivo, which means exterior, where cells are modified outside the body and then transplanted back in again. In this mode of gene therapy, genes are transferred to the cell grown in culture. Transformed cells are selected, multiplied, and then introduced into the patient. The use of autologous cells avoid immune system rejection of the introduced cells. The cells are sourced initially from the patient to be treated and grown in culture before being reintroduction into the same individual. Ex vivo gene therapy approach are most frequently applied to hematopoietic stem cells, which are relevant to blood and immunological disorders, cancer, and genetic disease that affect tissues and organs. Easily accessibly by blood cells. Among diseases potentially treatable with genetically modified hematopoietic stem cells is severe combined immunodeficiency, popularly known as bubble boy disease, whose history of treatment can somewhat be viewed as the history of gene therapy itself. So let's talk about the traditional treatment. Traditional treatment involves bone marrow transplantation early in life, replacing the hematopoietic cells harboring mutant genes with matching cells carrying healthy genes. In some gene therapy clinical trials, cells from the patient blood or bone marrow are removed and grown in the laboratory. The cells are exposed to the virus that is carrying that desired gene. The virus enters the cells and insert the desired gene into the cell's DNA. The cells grow in the laboratory and are then returned to the patient by injection into a vein. This type of gene therapy is called ex vivo because the cells are treated outside the body. To better understand, here is the self-explanatory schematic diagram for ex vivo transfer. So ex vivo therapy involves tightly regulated cellular manipulation in harvest cells. And now, let's talk about the benefits of ex vivo gene therapy. The advantage of ex vivo gene therapy is the ability to characterize the genetically modified cells extensively and select them for desired phenotypes before using them for therapy. The property of the cells may also be chosen to enhance the therapeutic potential.
Good day everyone, I am Jeden Flores Miranda from BPED 4C. Today I will going to discuss the second approaches for the transfer of genes in gene therapy, which is the vivo gene therapy. In this method of gene transfer involves the transfer of clone genes directly into the tissues of the patient. This is done in case of tissues whose individual cells cannot be cultured in vitro in sufficient numbers like brain cells, and or where reimplantation of the cultured cells in the patient is not efficient. Liposomes and certain viral vectors are employed for this purpose because of lack of any other mode of selection. In case of viral vectors, such type of cultured cells were often used which have been infected with the recombinant retrovirus in vitro to produce modified viral vectors regularly. These cultured cells will be called as vector-producing cells or the BPCs. The BPCs transfer the gene to surrounding disease cells. The efficiency of gene transfer and expression determines the success of this approach because of the lack of any way for selection and amplification of cells which take up and express the foreign gene. And that will be all. Thank you.
Good day everyone, I'm Adi Mateo from BPED 4C. In this video, I will going to discuss the target sites for gene therapy. Target sites for gene therapy. Therapeutic genes have to be delivered to specific target sites for a specific type of disease. In this table that I will show describes the list of such disease and their target sites for gene therapy. Target cells for gene transfer. These are the following disease and its target cells. Number 1, cancer. The target cells of cancer are tumor cells, antigen-presenting cells or APC, blood progenitor cells, T cells, fibroblasts, and muscle cells. Next is inherited monogenic disease, and its target cells are lung epithelial cells, macrophages T cells, blood progenitor cells, hepatocytes, and muscle cells. Number 3, infectious disease, and its target cells are T cells, blood progenitor cells, antigen-presenting cells or APCS, and muscle cells. Cardiovascular disease, and its target cells are endothelial cells and muscle cells. Next is rheumatoid arthritis. The target cells are synovial lining cells. And the last one is cubital tunnel syndrome, and its target cells is nerve cells. And that's all. Thank you.
Hello everyone, this is Shaina from BPED 4C, and to continue this report, we're going to tackle now with the advantage and disadvantages of gene therapy. So what are the advantages of using gene therapy? So gene silencing. In the case of an HIV infected person, gene therapy and gene silencing can be protect the patient from pain and suffering and before the disease is progresses. Now with the gene therapy, it is potentially used to eliminate and prevent hereditary diseases such as cystic fibrosis. It is also a potential way to treat heart disease, AIDS, and cancer. Additional advantages of the gene therapy is the advantages of RNAi include the high efficiency of gene knockdown, the ability to easily target the gene of interest as well as the stable and long-term silencing by expressing shRNAs. This makes more powerful tool that has been successfully applied to answer many questions in biology. So there are also risks, however, when using RNAi that need to be considered carefully. Plus, gene therapy replaces a faulty gene or adds a new gene in an attempt to cure diseases or improve your body's ability to fight disease. And gene therapy holds promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes, hemophilia, and AIDS. Now let's move with the disadvantage of gene therapy. So the novelty of gene therapy methods is one of the disadvantage. And it also says here the stimulation of immune response. The gene infected by a virus may cause immune responses due to the presence of the virus inside the body and the pathogenic potential of the viral vectors. And generation of genetic disorder due to the presence of multi-gene. The genetic material transferred may not necessarily enter the target cell. Even if it does, it may not be placed at an inappropriate place in the genome. And the also advantage of the gene therapy is the immunostimulation is defined as the unwanted overactivation of the immune system upon exposure to either pathogens or xenobiotics. So potential disadvantage of gene therapy is the poses number of risks. The way the genes are delivered and different vectors may prevent the following risk. So DNA mutations, the new gene might be inserted in the wrong location in the DNA, which might cause harmful mutations to the DNA or even cancer.